Okay. Well, we're actually getting now back more on top of things in terms of the timing and and where we are and everything. So I want to spend a little bit of time making sure we got the initiation and elongation of translation put into our heads. So let's spend a little bit of time just reviewing what I talked about last time. So you remember I said that, of course, translation occurs on messenger RNAs. And the messenger RNAs in prokaryotic cells have a sequence called the Shine-Delgarno sequence. That's very important for helping the ribosome to know where to start translating the protein. And the way that it works is that the Shine-Delgarno sequence has a sequence that's complementary to a sequence in the 16S ribosomal RNA that's in the small subunit. This is the small subunit here. Okay. So since it's complementary, they can form base pairs. And that base pairs actually helps to align everything. So this guy comes down here. You can see the base pairing is occurring right there. And as a result of that, this AUG start codon appeared, in this case, like right in the middle of this small subunit. Well, it's in the middle here where we see that this P site is going to be, the A, the P, and the E site, right? Okay, so that aligning was critical for that process. And so when we look at prokaryotic genes, we almost always see that Shine-Delgarno sequence. So the ribosome knows where to start, okay? That's a really important consideration. The ribosome knows where to start. We also saw in the process that after these guys form a complex, we have the tRNA that carries the initiator, 4-methionine. Remember, that's a methionine that's got a 4-mil group put on it, that comes in and forms a base pair with that initiating codon AUG. And after that, the large subunit comes in, and we have the entire complex then ready to go. So we've got the initiator transfer RNA with a 4-methionine in it. It's base paired in the P site to an AUG. The A site is open, and both subunits are present. Okay? Now, at this point, we say we're at the end of the initiation phase of translation. Okay? We haven't made any peptide bonds or anything, but we're at the end of the initiation phase because we're ready now to start putting those together. I want you to also remember what I said about that bond between the transfer RNA and the amino acid. I said that was very unstable. And I said there's a protein called EFTU that protects that. And so it's unstable in water. It's actually water that will break that bond. And so EFTU protects against that water getting access to the amino acid tRNA bond. Okay? Now, you might look on here. You're not going to see EFTU. So the one exception to the EFTU rule is actually the initiator transfer RNA. The initiator transfer RNA is brought in by something called IFTU. Or actually, it's called IF2, the number 2, IF2. IF2 functions just like EFTU does. It's just a different protein. Okay? So in any case, when a transfer RNA is floating around the cell, it's either protected by EFTU, or if it's an initiator transfer RNA, by IF2. All right. Now, at this point, as I say, we're at the end of the initiation phase. So now we move on to elongation of that message.
Before we move on,
yes?
The Shine-Delgarno sequence connects to a 16S or the 30S?
So it's in the 30S subunit, but it's in the 16S ribosomal RNA that's in that subunit.
Okay.
Right? So the 30S subunit contains the 16S ribosomal RNA, and it's that 16S ribosomal RNA that forms the base pair. Okay? One other thing I'll mention, and that is this. Notice that this initiator tRNA is different in another respect, in that it starts in the P site. It's the only one that starts in the P site. All the others will come in through the A site during the process of elongation. Okay. So actually, I was going to show you the so here's the Shine-Delgarno sequence. I said the consensus sequence is basically a GGAGG, okay? And you see the complement consensus sequence down here, CCUCC. That's what a Shine-Dalgarno sequence looks like. And here's the alignment of the AUG. Now, if you look closely here, you'll see that one of these guys actually starts with a GUG. And so in prokaryotic cells, there are occasionally start codons that GUG is actually read as an AUG. Okay? So there's a sort of an error-prone reading that happens with that. But that's one way. This is the summary of the Shine-Dalgarno sequence. And so it's, again, that's a consensus. It's not a perfect sequence. And we could imagine that this guy might have a translational efficiency that's very high because it's perfectly matching the consensus sequence. And this one might have a translational efficiency that's lower because it doesn't perfectly match that consensus sequence. And that would be correct. Questions? Okay. Now, I should also, I guess you could read it there, but this is not just the consensus sequence. It says consensus sequence, but this is actually the sequence of the 16S ribosomal RNA. So you can see there it matches what I said was the consensus sequence, CCUCC, pairs with GGAGG perfectly. Okay, well, anyway, the upshot is because the ribosome knows where the start codon is, it can start the process of translation that's necessary for all that initiation complex to come together. We see the same complex now flipped over. So this is the small subunit on the bottom, the large subunit on the top. We still have A, P, and E aligned as we did before. We see the A site is empty. So the A site must get an amino acid into that site carried by a tRNA. And so we see a tRNA come in that is complementary to this next codon. Notice that the A site has a codon sitting in it as well. And that transfer RNA that comes in has to base pair that perfectly. If it doesn't base pair it perfectly, then it gets removed. And this was one of the functions I said that EFTU had. EFTU looks for proper base pairing. If it doesn't see proper base pairing, then EFTU will remove that tRNA and let another one come in to do proper base pairing. Okay? Now, EFTU is an example of something that we'll talk about later in the term called a G protein. So I want to tell you what a G protein is. In fact, most of the factors, like the IF2 and the EFTU and all these various factors, are G proteins. That means that they bind guanosine nucleotides. So they bind GTP. And in the process of doing what they do, they hydrolyze GTP. So they convert GTP into GDP. So one of the things that's interesting about the process of translation is that the energy source for translation is GTP. Most of the cell stuff uses ATP as an energy source. But in the case of translation, all of the energy of translation comes from GTP. Okay? One difference of translation. All right. So here's the GDP that you can see that's been released. There's EFTU that has got that GDP. It's going to replace that GDP with a GTP at some point. You can see it came in as a GTP over here. Okay. Now, the next step in the process, we've got everybody loaded up here. We've got something in the P site. We've got something in the A site. Notice that these two amino acids are not joined yet. There's no peptide bond between them. So the formation of the peptide bond is the next step in that process. And the formation of the peptide bond is the responsibility of another ribosomal RNA. And that's a ribosomal RNA that's found in the 50S subunit, in the large subunit. That ribosomal RNA is called the 23S ribosomal RNA. Again, the S refers to the size. 23S is bigger than 18S, but it's smaller than 50S, right? The 23S ribosomal RNA is a ribozyme. It is. It's a catalytic RNA. And it catalyzes the formation of the peptide bond. So every peptide bond that's formed on the face of the earth happens as a result of catalysis by a ribozyme. That's kind of cool. Well, at this point, what's happened is we've made a peptide bond between the two. The dipeptide is transferred over to the newest transfer RNA, and the oldest transfer RNA is going to be squeezed out of the picture. Well, how does that squeezing out of the picture happen? Well, that happens as a result of action of another G protein, and the other G protein is called EF-G. EFG, very easy to remember. EFG is the machine that moves the ribosome down the messenger RNA. It literally pushes it down one codon. So you see where it is here. It's EFG using the energy of GTP pushes this guy down. So the AUG, which was in the P site, moves over to the E site, exiting. GCC that was in the A site now moves to the P site, and the A site becomes open again. Okay? So now we're going to see this process repeat. And it's just going to keep repeating every time there's a new transfer RNA that can come in and form a base pair with the codon that's in the A site. So every incoming transfer RNA is going to come into the A site first. And we're going to see this continuing forming of a peptide bond that's just going to keep pushing this thing further out here, and it's going to keep growing and growing attached to this tRNA that will come into here. Okay. So that process of elongation occurs until the the ribosome hits a stop codon. So a stop codon, remember in the genetic code that there were 3 codons that told the ribosome we're stopping making protein. I think you should know those sequences. UAA, UAG, UGA. Those 3 codons, along with the AUG, that's the other one that you should know. So AUG is the start codon. UAA, UAG, and UGA are the stop codons. Those are the ones that you should know the sequence of. And when they appear in the A site, something else happens. Okay? The something else happens is that there's no transfer RNA that's going to form a stable base pair with that. So it kind of bounces around for a while. The ribosome gets stalled, if you want to think about it that way. Well, the stalling of the ribosome allows something called a release factor to enter the A site, and the release factor looks to the ribosome kind of like a transfer RNA. But the release factor has something associated with it that's critical for ending the process. It carries one single molecule of something. What do you suppose it carries? What is that molecule that the release factor carries into the ribosome? Water. Because what water does is it breaks the bond between these 2 things. Remember, EFTU was protecting that all the other times. And once it got in the ribosome, there was no water in there. But the release factor brings water into the ribosome, cleaves that bond, and now the polypeptide is free to go. So the polypeptide might have started folding as it was being made. You can see a little bit of a fold going on right here. Okay? There are other possibilities, but it might have started being folded. Once it gets released, it can complete the folding process. And when it goes away, everything else falls apart. So that's the whole process of translation. Yes, Amy?
What happens to the water after the fact?
Well, the water actually becomes, it joins the bond. So it's a hydrolysis reaction. So part of the hydrogen part goes to one side, the hydroxyl part goes to the other side. So it becomes a part of the tRNA and a part of the protein. Okay, now that's a very intricate process. And I don't just throw all this intricacy at you and say, okay, well, just now you got something more to memorize. I mean, I think that you should know how the process works. But I show you this to impress on you the incredible nature of this nano machine. Okay? This is a nano machine. Engineers spend their careers trying to design a nano machine that can, for example, at the nanoscopic level, move or flip or do something fairly simple. This nano machine, okay, has gone through all those steps that I just described to you it gets it right, all right, and it makes it at the rate of about 30 amino acids per second. Now, that's not 1,000 like we see with DNA replication, but DNA replication is aimed at doing something else. This guy's making proteins for the cell. It's doing all these factors. It's doing all this hydrolysis. It's making sure it's got the right base pairs in there, and it's doing it at about 30 per second. That's really remarkable. Okay? Engineers who want to make nanomachines can't make something this intricate yet. Okay? We're a long ways away from making things this intricate in the laboratory. Okay. Questions, comments? My quiet class. Okay. I show you this slide to not give you a whole bunch of structures because that's not the important point. But one of the things I like to impress on students is the importance of understanding of some of these processes. Okay? You saw a fairly intricate process for translation, and you saw that translation required amino acyl tRNAs, right? There's an amino acyl tRNA structure where the rest of the tRNA is here, and there's the very last base on that amino acyl group. So we could imagine that cloverleaf structure going all the way out here. And here's where the amino acid would be attached, right? If I look at that, what do I see? I see that I can make structures that look like that attached nucleotide, and I can confuse ribosomes. So this is something like where competitive inhibition that we talked about, where the substrate resembles where the inhibitor of an enzyme resembles a substrate. Here we're making a molecule, in this case puromycin, that resembles a part of a transfer RNA. What happens is the ribosome says, oh, you're a transfer RNA. I will let you in. And puromycin gets into the ribosome, and it gums it up. So many antibiotics work by gumming up translation. Many antibiotics work by gumming up translation. If you've ever had to take chloramphenicol when you were a kid, okay? Chloramphenicol is an antibiotic that gums up translation. Puromycin is another one that gums up translation. Antibiotics are important for us to understand in terms of strategies for how they kill, right? I could design something that would kill all cells. Let's imagine I made something that chemically degraded DNA as soon as it got into the cell, right? That would kill any cell that had DNA because cells need to have DNA, right? But if I design something that kills only something that occurs in bacteria, I've made a selective antibiotic. And so what most people think of in terms of antibiotics, or what I refer to, are selective antibiotics. You want something that kills nasty bacteria, but you don't want something that kills your cells because that's considered not a good career move, right? So if we can find things that are unique to bacteria, the bacteria need to be alive, and we target those processes, then we have a way of making a selective antibiotic, which is the best kind of antibiotic that we can have, right? So how does translation, Kevin, you've been telling us that translation is the same in prokaryotes and eukaryotes. Well, there are enough differences in the structures of the ribosomes within the prokaryotic cells that's different from our cells that we can actually design things that will selectively gum up prokaryotic ones and not affect eukaryotic ones. So chloramphenicol is a real good example. Okay? It will only affect prokaryotic ribosomes and consequently only kills bacteria. It won't have any effect on our cells. Kind of cool. So a lot of antibiotics work in that way. Questions about that? Okay. Let's talk about eukaryotes. So overall, the process of translation in eukaryotes is similar to that in prokaryotes. The ribosomes, as I said, are a little bit different in structure. They're bigger, for one. All right? So whereas the prokaryotes had 30S and 50S subunits for the small and the large, respectively, the eukaryotic will have 40S and 60S. They're considerably bigger, with an overall structural size of 80S. Okay? So again, the S's don't linearly add, right? But 80S being the sum of the two. They each contain ribosomal RNAs, like the prokaryotic cells. Okay. So the small subunit contains a ribosomal RNA called the 18S. The 18S. Now, one of the differences we see in eukaryotic cells is there's no Shine-Delgarno sequence. There's no Shine-Delgarno sequence. There appears to be some structural information about where the start codon is, and I'll talk about that in just a bit. But there's no Shine-Delgarno sequence. So the 18S, which is the rough equivalent of the 16S from the prokaryotic cells, doesn't have a sequence complementary to the messenger RNAs that are found in eukaryotic cells. Okay? So there has to be a different strategy for finding where that first AUG is. Okay. The large subunit contains several ribosomal RNAs, and I won't even bother you with, well, I will with one of them, with all the sizes. Okay. The one that matters is what's called the 28S ribosomal RNA. That's the equivalent of the prokaryotic 23S because it's the 28S that has the ribozyme function. It forms the peptide bond. So like in the prokaryotic cells, it's found in the large subunit, okay? And that's what its function is. Now this figure shows a difference that I talked about in the last exam relative to the structures of the messenger RNAs that are found in eukaryotic cells. You remember it has a 5' cap, which has that unusual 5' to 5' bond at the 5' end. And one of the things we're going to see about that cap is not only does it protect against nucleases, which I talked about last time, but it also plays a role in translation. Okay? At the 3' end of the eukaryotic messenger RNAs, we have the poly-A tail that I talked about, and that's a stretch of anywhere from 2 to 300, or this is 50 to 100, but up to 2 to 300 poly-A's that can be present on the 3' end of that message. Within there, we have a region that's translated, and we also have a region called the untranslated region on the 5' end and the untranslated region on the 3' end. Now, I show you that to remind you that the AUG is not present on the end of the messenger RNA. It's the same as in prokaryotic cells. It's not present on the end there either. Yes? Why are the untranslated regions not spliced out? The reason is because they actually have functions. Okay? So they play roles in efficiency of translation and also in some cases in recognition of proteins that help to say where the start codon is. Okay? It's a reasonable question. Yeah. One of the things in prokaryotic cells is we see that the untranslated regions are usually fairly small. So maybe, it might be 20 nucleotides or 30 nucleotides on the end, and the same thing at the other end over here. Okay? In eukaryotic cells, this could be a couple hundred base pairs. And so the problem for eukaryotic cells is the eukaryotic cell has got to know, where do I start? Where's that AUG that I'm after? Okay? There's a couple of things that eukaryotic cells appear to do to be able to find that. One is that it's very common in eukaryotic cells that the first AUG is the right one. So then what the cell has to do is just simply go sliding down, find the first AUG, and say, oh, this must be it. I'm going to start translating. The ribosome does that, right? So I'm going to find that AUG, and I'm going to start translating. So for a reasonable approximation, that works reasonably well. Remember that eukaryotic cells don't have operons. So they don't have to worry about another protein over here they've got to translate. They've only got to translate one, and they've only got to translate it using the first AUG. Okay? So it's not as complicated of a system as it is in prokaryotic cells that will have multiple genes on a given messenger RNA. Okay? All right. Another thing that they appear to use, and we're not completely sure how this works, is that there are a few bases immediately around the AUG in eukaryotic cells that appear to be fairly common in all genes. They're not quite as conserved as the Shine-Delgarno sequence in the prokaryotic cells, And we don't know if something binds to them or recognizes them. We're not quite sure what role, if any, they play. But there may be some sequence information around that AUG that helps eukaryotic ribosomes to know where to start. Okay. Now, the last thing I want to mention is that role of the cap and the poly A in the process of translation. Now, this looks like a figure like, oh, my God, Ahern, you're not going to do this to us, are you? No, I'm not. I'm showing you something that I want to keep very simple. What I'm showing you is that same messenger RNA in a little different view. Here's the 5' end. That's where the methyl G is. That's where the cap is down here. Here's the messenger RNA running all the way over here to the poly A tail at the 3' end. So if we follow the green and the purple, we're seeing the messenger RNA. So what's all this other crap in here? Well, here's what some of that crap is. There's the small subunit, the 40S subunit. Okay? Here are a bunch of things that say EIF3, EIF34A, EIF4G. You're not going to memorize those, by the way. Okay? See all these things? Well, in prokaryotic cells, we had initiation factors. Remember IF2? IF stands for initiation factor. So this is an initiation factor that is helping the ribosome to assemble, just like the prokaryotic system did. However, they've got different names. E stands for eukaryote. Eukaryotic initiation factor 413B, right? The number doesn't really matter. The point is that these initiation factors help the ribosome to assemble, and they recognize, in this case, the cap, and or they recognize, in the case of this one, the poly A tail. So both the cap and the poly A tail play important roles in the initiation process. What do all these others do? Well, I'm not going to go through functions with you for them, because that's more than we need to do in this class. But you can see that the process of initiation uses a lot of proteins to help assemble that ribosome. One of the questions people have is, well, why make this circle? What's the advantage to making a circle like that? And it's thought that the ribosome, once it gets started along here, okay, if another one starts following it behind, this circle forms, because at the end of this start, this whole thing's going to fly away, right? So if another one starts while this one is going along, this circle will reform, and the ribosome, when it's done over here, doesn't have to travel too far to get back over and start again. So the circle may help keep things in close proximity. That's one thought about why that forms a circle. Prokaryotes don't do this. They don't form a circle at all. Okay? Eukaryotes, we know they do that. Okay. Questions on that? So I'm not asking you to memorize any structures there. I just want you to know the cap plays a role. I want you to know the poly A plays a role. I want you to know that eukaryotes have a circle and prokaryotes don't have a circle. Okay. Let's talk about a couple things fairly loosely related to what I've just been talking about. Okay? We talked about zymogens, right? Everybody remember zymogens? Inactive forms of enzymes that were made. Okay? And technically, they're inactive forms of proteins that are made, because I'm going to show you a zymogen here that doesn't catalyze a reaction but plays an important role in your body. And that's the hormone known as insulin. Insulin is a hormone. It's what we call a peptide hormone, which means it's synthesized using ribosomes. It's coded for in your DNA. And insulin is a protein that goes through several modifications to become the what we call mature insulin that your body uses. Okay? So here's the DNA going to messenger RNA, that's transcription. Messenger RNA going to protein, that's translation, right? And here's the precursor of pre-pro insulin. Don't get hung up with the names. Pre and pro mean that they're unmodified forms of insulin. So there's a couple modifications that happen. Okay? Modifications that have to happen to insulin include formation of disulfide bonds. So we think about that folding process. Remember when we said folding happens, then two cysteines can be brought into close proximity, And when that happens, they can make a disulfide bond, and that's a strong bond. Okay? Well, that turns out to be pretty critical for insulin, because look what happens to insulin along the way. This single polypeptide chain gets its ends cut off. And when it gets its ends cut off, if those disulfide bonds aren't holding everybody together, this piece goes flying away. You wouldn't have insulin. Okay? Now, this is a real good example of covalent modification of a protein that's necessary for the protein to function. This will function as insulin. This will not. Okay? We'll talk later in class about what insulin does and the significance of insulin. All right? But this is a prime example of a post-translational modification. It's called post-translational because it's happening after the process of translation. After the protein has been made, it gets modified. Yes, sir?
Do we find the mutation to the sequence coding for the removed portion of the program, something less deleterious?
That's a very good question. As far as I know, what you said is correct. Mutations that happen out here in parts of the protein that don't matter are less deleterious. There's a limit, though, because the proteins that recognize these things, if we mutated the recognition site for the deletion, then that would have a very deleterious effect. But if we had something up here, for example, that doesn't appear in the mature protein, yeah, you bet. That would have very little effect on the insulin. Good question. Okay, others? Okay. So now I want to tell you something else that's kind of cool. Okay, you said there's 20 amino acids that go into proteins. And I said they occupy about 99.99999% of all proteins. But there are, as you know, whenever there's a rule in biology, there's almost always an exception. The only exceptions we've seen, only things we've seen that don't have exceptions are DNA polymerases always work only in the 5' to 3' direction. RNA polymerases always only work in the 5' to 3' direction. And translation always only works in the 5' to 3' direction. So that 5' to 3' direction is a pretty absolute thing in how these various enzymes work. Well, it turns out that the rule about 20 amino acids is not completely absolute. There's another amino acid that occasionally appears in proteins, and it's called selenocysteine. The structure of selenocysteine is shown on the screen, and as its name would suggest, it contains a selenium. There's the selenium. And it's related to cysteine. The way it's related to cysteine is the selenium replaces the sulfur. So if I had cysteine, that would simply be a sulfur. Since I have selenocysteine, that's a selenium there. Okay? So what happens here? Well, it turns out that selenocysteine gets into proteins not by a covalent modification, not by something coming along and pulling out a sulfur and putting in a selenium, but instead it gets actually put in during the process of translation, okay? Which means it somehow has to be coded for, because translation requires tRNAs that have coding. So something means that this guy has got to be put onto a tRNA. It means that some tRNA has got to be read by the ribosome so that it takes it, and then only then will this get built in during the process of translation. The cell uses a very odd trick to do this. It makes a special tRNA that is complementary to one of the stop codons. It's complementary to one of the stop codons. This guy gets to go in where a stop codon would otherwise appear. Okay? And instead of the translation stopping, this gets put in and translation continues to a different stop codon. Now, this doesn't happen real efficiently. Sometimes the protein stops instead of getting the selenium in which case you've just made a protein that's of no use to the cell. Other times, it makes this, okay, and everybody's fine. Okay? Now, there are several enzymes that we have that contain, and that every organism has that contains selenocysteine. So this is not a trivial thing. There's not very many, but there are a handful. Okay? So one is an enzyme called thioredoxin reductase. You don't need to know this. I'm just telling you this, okay? Thioredoxin reductase. Thioredoxin is a molecule that's very important to act as an antioxidant. And it has a cycle that goes through of oxidation reduction. The reductase catalyzes the formation of the reduced form of it. Thioredoxin reductase have this molecule in there. Okay? Another common enzyme that contains this molecule is the thyroid-modifying enzymes that you have. You have enzymes called deiodinases that modify your thyroid hormone. That's the hormone that has iodine in it. Okay? Every known deiodinase, for some reason, contains this guy right here, okay? Which makes it doubly odd, right? Because this is the only place in the body where we find selenium. Selenium is a poison. You take selenium supplements in vanishingly tiny quantities. Virtually nobody should take selenium supplements. But if you ever needed a selenium supplement, your doctor would give you something that was in extraordinarily tiny quantities because selenium is poisonous. So the only place you see selenium is here. The only place you see iodine is in those thyroid hormones. Kind of cool. Okay. Yes? Selenium, actually, selenium is in Brazil nuts, and there's actually an odd form of methionine that contains that, yes.
Okay. So is it possible to, like, be poisoned?
No. No. So, no, you would have to eat a lot of Brazil nuts. When I say poisonous, I'm talking about, you know, something that would be an abnormal amount. I wouldn't recommend that you stop eating Brazil nuts. But I'll tell you what's in Brazil nuts, and it's found almost only in Brazil nuts as far as I know. And that is methionine is the other amino acid that contains sulfur, and it contains a sulfur between two carbons. Brazil nuts have a modified form of that methionine that has a selenium between those two carbons. Yeah. And I don't know of anything else that has that, which is kind of cool. Yeah. That's not something a lot of people know. Okay, other comments, questions? Okay. Well, we're going to get through translation here, so let's do that. And then I have something for you. Okay. So the last thing I want to talk about is something that's very important for proteins. We've talked about the importance of proper folding of proteins. We know that if proteins don't fold properly, they can, in some cases, cause awful diseases like mad cow disease or Creutzfeldt-Jakob syndrome, which I think I talked about. That's a human disease where that equivalent protein in humans misfolds and causes severe dementia and very rapid death. Okay? So the folding of proteins is important. The proper folding is important. When people first started studying bacterial cells, they discovered something interesting. They discovered that bacteria, which live in our gut, for example, E. coli is the one that they study, really like to grow at about 37 degrees centigrade. It's our body temperature, 98.6 Fahrenheit, right? And that if they took those bacteria and they heated them up to a little bit higher temperature, like about 41 or so centigrade, okay, it's about 7 or 8 degrees, like a pretty good fever that you might have, okay? The bacteria started making other genes. They started making other genes. And they knew that they were making those other genes in response to the heat. The heat was causing a problem. Okay? Of course, if you heat them up high enough, you'll kill them because you'll completely denature. But if you heat them up just a little bit, 41 to 42 degrees, they started making these other proteins. And they classified these proteins as heat shock proteins. Okay? Heat shock proteins. And those heat shock proteins were interesting because you could look across a wide variety of bacteria and you would see they would be made. And when you started looking in eukaryotic cells, you could see the same sort of thing. Well, they later came to realize that the cells made these proteins not just during heat shock, but heat shock was a good way to do it. Okay? But they made them in other ways for other purposes. We tend to make these heat shock proteins most of the time. And these heat shock proteins, when they analyzed them in bacterial system, they're called GRO-S and GRO-L. And what these heat shock proteins were doing was that they were helping proteins to fold properly. Now imagine if you add heat to a protein, you might denature it a little bit, right? If you wanted it to fold properly, you'd like to have something that helps it to fold, and that's what these heat shock proteins were doing, the GRO-L and the GRO-S. Okay? So I'm going to tell you a little bit about how they work and so forth. Okay? Well, in our cells, we have the equivalent of these two that's being made all the time. And they're being made all the time to try to help proteins to fold properly. Now, I should point out that misfolding of proteins can happen and probably happens all the time and has no effect whatsoever. So it's not like if I say we had a single misfolded protein, you're going to get this deadly disease and die. That's not what's going to happen. Okay? But you do want to reduce the incidence by which misfolding happens. So let's imagine that I'm a cell, and I have a ribosome that's moving down a messenger RNA, and it's translating. Remember that we have hydrophobic and hydrophilic amino acids, right? Where do the hydrophobic amino acids end up in a folded protein? Inside of the cell, if that protein is dissolved in water, right? Because the hydrophobic amino acids avoid each other, right? Okay. Imagine I've got this messenger RNA that runs along here, and I've got this ribosome trucking along over here, and I've got a ribosome right next to it trucking along making the same thing. This one's a few amino acids ahead of this one. Everybody envision that? It's moving along, and it hits the stretch where this guy starts hitting a bunch of what will be hydrophobic amino acids, right? It gets this. The hydrophobic amino acid starts coming out of here. What does the hydrophobic amino acid want to do? It wants to avoid water, right? The hydrophilic one that's following it hasn't started coming out. So it gets over here, and now this one starts putting out hydrophobic, and this one's still got some hydrophobic over here, right? Do you suppose these hydrophobics on each one might interact with each other? They might, okay? So what happens is for proteins that need help folding, they actually get made into an organelle we call the endoplasmic reticulum. And the protein gets squirted in there and grabbed by these things. What these things do is very simple, surprisingly simple. They sit there. They just sit there. The protein sits inside of them, and the protein is in this chamber that it can't interact with. And so the protein gets a chance to fold on its own. Instead of interacting with this protein over here, it folds on itself. So this guy allows the proper folding to occur, and after that proper folding occurs, it gets released as a properly folded peptide. Yes, Andrew?
Is this happening when the protein is coming out of the ribosome?
That's correct. As the protein is coming out of the ribosome. Yep.
How is this thing related to the chaperone?
This is a chaperonin. So I haven't gotten to that. You're ahead of me. So these we now call chaperones or chaperonins. That's what they are. So instead of calling them heat shock proteins, in our cells we call them chaperonins. Okay? So if you hear that term, either chaperones or chaperonins, which refers to the individual proteins, that's what we're talking about. This is a bacterial system, but it's very equivalent to ours. It's just called GRO-L, GRO-S. Did you have a question? That was your question? Okay. Clear as mud? Yes? You can have some out in the cellular cytoplasm, but most of this is happening actually in the endoplasmic reticulum. Yeah. Okay. Well, you guys have been a patient group. We had the computer down for a couple days, so we didn't get to sing a song. So I have two songs for you today, and they're both easy to sing. And if you sing loudly, we might have extra credit on the next exam, right? So they're very easy. The first one, I have to ask you to stand up and put your hands on your heart, though, because there's a very important reason why. Hands on heart, guys. Wrong one? Okay. This is the national anthem of biochemistry. Everybody, that's like the national anthem. You stand, you put your hand on your heart, and we're going to sing this loudly. It's very easy, and the reason it's the national anthem is it's sung to the tune of, not that one, it's sung to the tune of America the Beautiful. Oh, beautiful with RNA to make the peptide bonds. You hold tRNA so it can pair up with codons. The ribosome, translate mRNA, initiate and translocate from start to UGA. Okay, now you can sit down, and we'll do our other song, which is to the tune of Old King Wenceslas. It's called Good Protein Synthesis. And it goes, amino acids cannot join by themselves together. They require ribosomes to create the tether. All the protein chains get made according to instruction, carried by an mRNA in peptide bond construction. I screwed that up, didn't I? Small subunit starts it all with initiation, pairing up to RNAs at the docking station. Schindel-Garnos complement in the 16 S's lines the AUG up so synthesis commences. Elongation happens in ribosomic insides where our RNA creates bonds for polypeptides. These depart the ribosome, passing right straight through it in the tiny channels there of the large subunit. Finally, when the sequence of one of the stop codons parks itself in the A site, synthesis can't go on. P site RNA lets go of what it was holding, so the polypeptide can get on with its folding. Okay, guys, see you Friday.
Good afternoon.
You too. Take care.